So protein folding is a process that really is central to life itself. Although some proteins in some small regions within larger proteins do function in a disordered or unfolded state, the vast majority of them function in the folded state, and they require the folded state to achieve their catalytic or structural specificity. So this is a tremendously important process biologically. It's also rather complicated and rather interesting, and so it's been the subject of study by people with many different kinds of technical expertise, from sort of hardcore statistical physicists and other kinds of physicists to traditional biochemists. Proteins, of course, life exists in water. Proteins fold in water, and then the folding of proteins is intrinsically dependent on the interaction of the polypeptide with water and the thermodynamic properties of water. So in addition to the importance of it, it provides a very deep insight into the thermodynamic, how life processes are influenced by the thermodynamic properties of water. So it pulls together a number of different things, a wide variety of different things that we have discussed in the treatment of thermodynamics in this course. It was shown by someone named Chris Anfinson, who is a professor at Johns Hopkins, in the 1950s, that you could unfold a protein using what are called chemical denaturants, urea guanidinium hydrochloride, and that it would spontaneously then refold. So he was able to demonstrate spontaneous refolding of proteins subsequent to complete unfolding in vitro. He won the Nobel Prize for that. And that research was conducted sort of through the course of the 50s and then went on from there. By the early 60s, people were studying this quantitatively using a number of different methods, including using something called microcalorimetry, where you can actually measure directly the amount of heat that is required to unfold a protein. Doing such studies, sort of capitalizing on the methods developed by Amundsen to look at chemical denaturation, and then using those microcalorimetric instruments. Someone named John Brantz, the University of Amherst, predicted in 1964 that protein should unfold at low temperature in addition to unfolding at high temperature. What Brantz had done is he had measured quantitatively the entropy and the enthalpy of the folding process at different temperatures, and he could see that there were wild changes in those parameters. And when he looked at their trends with temperature, he predicted that some proteins ought to actually unfold at low temperature. It's well known that they unfold at high temperature. Every time you cook an egg, you are heat denaturing, thermally denaturing the albumin in the egg. From people who had been working in biochemistry for a long time before the 1960s knew that in general, when you heat up a protein, eventually it reaches a point where it loses its stability in the folded state and spontaneously unfolds. So in 64, Brandt proposed that it should also, some proteins should also unfold, again, based on measurements, making quantitative measurements at temperatures where the protein is stable, and then looking how the changes in the entropy and the enthalpy of that process, varied with the change in enthalpy and the change in entropy varied with for folding varied with temperature. He said that those variations are such that at some point, sort of everything should reverse itself, and going to lower temperature should also induce unfolding. So in 1986, Peter Privilov, working at the Pushchino Institute of Protein Research in the Soviet Union, presented data in the Journal of Microbiology confirming this prediction, and he demonstrated that something called apomyoglobin. Myoglobin is a single chain equivalent of hemoglobin that is expressed in certain organisms and actually at certain times in the developmental process for mammals. He showed that apomyoglobin, which means myoglobin without the heme group, does experimentally unfold at low temperature in addition to unfolding at high temperature. So this is rather remarkable that inputting heat at one temperature can drive protein folding, while inputting heat at a higher temperature drives protein unfolding, which is exactly the reverse reaction. So you have heat having a fundamentally different role the input of heat has a fundamentally different role when proteins are examined at higher temperature compared to when they're examined at lower temperature. So this is rather remarkable, and it ultimately reflects very important things about the thermodynamic properties of water. If you can understand in a qualitative way what's going on here, all thermodynamic phenomena many thermodynamic phenomena in water become much easier to understand. And so that's why we're going to spend a little time looking at this. So this slide presents what I call an oversimplified view of protein folding. Essentially, the unfolded protein is in a large collection of different conformational states. I'll say more about that in a moment. When it folds, it adopts a well-defined conformation. And examples of the kinds of conformations it tends to adopt are shown elsewhere in the PowerPoint deck. And of course, folded proteins tend to be rich in regular secondary structures for the reasons that are outlined elsewhere in the PowerPoint. So the folding process is a conformational reaction. It can be treated thermodynamically like any reaction. In this case, it's a unimolecular reaction between the folded and the unfolded states, or vice versa. You can write down an equilibrium constant for folding, which would be the concentration of folded over unfolded. That would be the reciprocal of the equilibrium constant for unfolding. The delta G for folding will be negative RT natural log of K folding, which would be negative the delta G of unfolding. And so the basic thermodynamic formulas that we used earlier can be directly applied. So in the unfolded state, again, this is sometimes called a random coil state. Whether it's really entirely random, people debate, but it is pretty damn random compared to the folded state. So in the unfolded state, the polypeptide backbone is randomly, or more or less randomly, sampling the allowed Ramachandran space, whereas in the unfolded state, pretty much the dihedral bonds of the polypeptide backbone can adopt any of the allowed conformations in the Ramachandran plot. And so essentially in the unfolded state, every single amino acid C-alpha group is going to be exploring a C-alpha atom. The bonds attached to the C-alpha to every single C-alpha, the amide bonds attached to them will be exploring, randomly moving around in the allowed areas of the Ramachandran plot. Once it folds, in this case, most of the residues here are helical. They now no longer are exploring that large conformational space. The helical amino acids are all going to be constrained to some very narrow region in the Ramachandran plot represented the alpha helix. Basically, every amino acid in the folded protein will now have a conformation characterized by sort of one point or one very narrow zone within the Ramachandran plot. So all the conformational freedom reflected by the totality of the allowed areas in the Ramachandran plot is lost when the protein adopts the folded conformation. And so it goes from a high entropy state, again, very high entropy state, because every amino acid to a first approximation in the unfolded protein is exploring all these Ramachandran areas. So it's many different possible microstates. And then upon folding, it's going to be constrained to a very narrow set of angles, approximately a point when you view things from this scale. And so there's an enormous loss of conformational freedom of the backbone. Similarly, the side chains in the unfolded state, the side chain dihedral angles are going to be randomly sampling the allowed rotamer states. And when the protein folds, the side chains, certainly in the interior, are all going to be constrained to a single rotamer state. And so the side chains also will undergo an enormous loss of conformational entropy. So again, the unfolded polypeptide, in the unfolded state, the polypeptide backbone randomly samples allowed Ramachandran space, while side chains sample all rotamers. And there's an immense ensemble of different backbone and side chain configurations. In the folded state, in contrast, the polypeptide backbone and side chains adopt defined conformations. And so this is a comparatively tiny ensemble of closely related backbone and side chain configurations. There's an enormous loss of configurational entropy when the protein folds, because, of course, the second law has told us that all physical systems proceed spontaneously to their highest entropy state. Proteins, in the absence of some other thermodynamic effects, proteins are going to tend to go into the unfolded state because it's much higher in entropy. Okay, so the protein folding process, as I said earlier, has been incredibly widely studied in many different ways. This is one, it's easy to find things on it, to read up on it online. Most of what I found prowling around, was at least reasonably accurate and realistic. The bottom line, and so one sort of a set of ideas about folding is summarized here. I think the very hardcore biophysical studies show that sort of everything depicted here does happen to some extent in some proteins. Whether it's exactly any one of these ways in any given protein is rather well, there's certainly some controversy. For the purpose of this, what we're interested in here in this presentation, in this course, this is not terribly important, but the general idea is that proteins start in the unfolded state, again, randomly sampling an enormous number of configurations. Many studies show that they tend to go to intermediate states that are condensed, where you have minimally more interaction between the different parts of the chain, perhaps formation of some secondary structures, and then later these intermediates condense into the final stably folded structure. Some of these intermediate states are what are called molten globule-like intermediates. Again, this isn't terribly important for this course, but this is an interesting sort of state of the protein. It's a collapsed state where actually a lot of the secondary structure has formed, but the tertiary structure of the protein is still not there. So again, for the purposes of this course, this material is not terribly important. Just want to have you give you a sense that there is a lot more known about it than we're going to talk about today, and that there is a sequential nature to the process in practice. But nonetheless, we can still look at the equilibrium constant, right? These are transient species. When we're looking at the folding process, we can still look at the equilibrium constant, or the energy relating the unfolded state to the folded state. And so, again, the unfolded states are sometimes called random coil, stochastically sampling Ramachandran and allowed phi space. The side chains are similarly, as the previous slide indicated, sampling many different rotamer states. There are negligible interresidue interactions, and it's highly extended. Art talked about some of these in the hydrodynamics module of the biophysics course. Some of the relevant polymer statistics things were discussed, and this state is reasonably random coil-like. Okay, and now and this is just another view of this sequence of events, and if you're interested in reading more, the websites linked here have pretty informative treatments. Okay, so however, the point of this screencast in this segment of the course is to understand what I told you about earlier, how and why proteins can unfold both with increasing temperature or input of heat, or decreasing temperature and removal of heat. And so we're going to start our effort to understand this by looking at the folding process at nearly physiological temperature. So this is now, we're looking at what would happen at a single temperature, let's say roughly 37 degrees. I should say the numbers that I'm going to show as I go through this presentation and the exact magnitudes of these things are sort of arbitrary. I've made these numbers up, frankly, but they do realistically reflect roughly the kind of numbers you see in actual proteins, and the relative direction and the relative magnitudes of all the effects do match closely, accurately, what is seen when you actually quantitatively study protein folding. So I should also note that this presentation here, it matches what Schultz and Schirmer discuss in their textbook. They treat the folding process at one temperature, at a temperature where the net effects are as they are shown here. So what are we seeing here? So we're going to look at two things. We're going to look at T delta S, basically the entropy of unfolding. Of course, the contribution delta G is equal to T delta H minus T delta S. And so effectively increasing T delta S means decreasing delta G, and so higher entropy means lower delta G. This is favorable. So the favorable entropic terms are going to be pointing upwards. Delta H, on the other hand, delta G equals delta H minus T delta S. So increasing delta H means increasing delta G. So the positive direction here is unfavorable in terms of unfolding, right? So we're going to separate out the enthalpy of unfolding. A higher enthalpy means positive enthalpy means heat has to be put in to the to drive the reaction. That means heat has to be taken out of the thermal bath, and that is, of course, entropically unfavorable. So positive enthalpy means heat being put in means unfavorable free energy. Positive entropy, on the other hand, means entropy gain, like the second law tells us will happen in spontaneous processes, and that means negative delta G or favorable. So basically, favorable here is down, favorable here is up. And again, we're looking at unfolding. If we were looking at folding, the magnitudes of these things would be the same, but the signs would flip. So let's look first at the entropic contributions. So as we noted, as I explained in the earlier slide, the unfolded protein adopts an enormous number of different conformational states compared to the folded protein. So when the protein unfolds, there is an enormous entropy gain. And this can be the equivalent of hundreds of kcals per mole at physiological temperature. So you get a gain in the entropy of the polypeptide or the chain configuration when the protein unfolds. On the other hand, you do have an entropic loss caused by the greater degree of solvent interaction. When the protein unfolds, although its bond angles are now flapping around much more wildly, water is interacting with much more of the, with much greater surface area, with much greater number of atoms from the protein. When the protein unfolds, the formerly buried atoms in the core are now exposed to solvent. The interaction with the solvent constrains the solvent. And so that constraining activity is stronger when it's interacting with hydrophobic groups than it is with hydrophilic groups. If you dig deeply into the literature, there are some complications there, but to a first approximation, especially at lower temperatures, the constraining effect of the hydrophobic interactions is greater. But the hydrophilic interactions can constrain the water also to some extent. So the bottom line is when the protein unfolds, you have an enormous increase in the entropy of the protein chain, the backbone and the side chains, but you have a decrease in the entropy of the solvent. That entropy gain and entropy loss will add together, will offset one another, and you will get a net, so you'll have to add these two effects together to see the net change in entropy of the system upon unfolding. And around 37 degrees, that is typically positive. That means it is dominated by the entropy of the polypeptide chain. You are getting more increase in entropy from the configurational freedom of the polypeptide chain than you are losing entropy from the restriction of the solvent. So now let's look at the enthalpy end of this. When the protein unfolds, you have to rip apart all the nice hydrogen bonding and van der Waals interactions between the atoms inside of the protein, right? So you have to put in heat, to put in energy, put in heat to overcome the favorable potential energy involved in those interactions, right? These are basically all different kinds of electrostatic interactions, van der Waals interactions, or favorable electrostatic interaction, hydrogen bonds, favorable electrostatic interactions. We have to pull apart all those favorable electrostatic interactions. So to unfold the protein, we need to put heat or energy in, right? And so it's a positive delta H. That means energy has to come out of the environment and go do the work of pulling apart the polypeptide chain. On the other hand, as the chain is pulled apart, those atoms that had been in the core are now interacting with the water. That is what is constraining the water. The water molecules are constrained because they have a favorable enthalpic interaction. There's a favorable electrostatic interaction energy of the water with the newly exposed atoms from the protein. And so essentially heat is released, right? Heat is released as the water is electrostatically attracted to the protein atoms. That constrains the water, which is entropically unfavorable, but there's a negative delta H, or a heat release, a favorable heat release. So the net effect on the enthalpy then we'll have to get by summing the energy that we need to put in to pull the protein chain apart. To take the difference between the energy we need to put in to pull the protein chain apart and the energy released when due to the increased solvation, and at around 37 degrees or lower temperature, the amount of energy you need to pull the chain apart exceeds the amount of energy you gain from the hydration. And so there is a net positive or unfavorable delta H for unfolding. To get the delta G for unfolding, we have to take delta H minus T delta S, right? So as it's schematized here, delta H is higher than delta S, so delta H minus T delta S would be a positive quantity, positive delta G. Positive delta G is unfavorable. Protein unfolding is unfavorable, which means that protein folding is generally favorable for most proteins at 37 degrees. So Schultz and Schirmer discuss this, present an analysis that is exactly follows this development. They discuss it in a little bit more depth. And so if you need clarification on this, you can read that material in Schultz and Schirmer. Okay. So key points. Notice that we had huge entropy and enthalpy. We had huge offsetting enthalpy effects that added together to give a smaller net enthalpy difference. And similarly, we had huge offsetting entropy effects that added together to give a relatively small entropy difference. But to get the free energy difference, we now have to look at the difference between these terms, and that is an even relatively smaller quantity. And so it turns out that that quantity, which determines the net stability of proteins, is typically relatively modest. The folded state of most proteins is only modestly more stable than the unfolded state, even at physiological temperatures. The delta G of folding is typically about -4 to -8 kcals per mole. If you think about it, that will correspond to an equilibrium constant on the order of 200 to at the low end to more like 1 million at the high end. So the stability, importantly, is determined by a relatively small difference between large offsetting enthalpy and entropy effects. And the enthalpy, right? We have offsetting enthalpy and entropy effects that will, have to be summed together, or you have to take the difference between them to get the net stability of the protein, the net free energy of folding. The enthalpy and entropy effects themselves are determined by differences between even larger offsetting contributions. And so the bottom line is solution thermodynamics can be complex. And what we're going to see is as the temperature of water changes, the sign of the magnitude, of the hydration effects is going to change a lot, and it's going to change enough to sort of reverse the balance between these things. And that's where we're going with this. So the hydrophobic effect is highly temperature dependent, as I just said, which creates even more complex thermodynamics behavior and is ultimately responsible for the fact that both increasing temperature and decreasing temperature can denature proteins. So the main thing going on here is that at low temperature, water moves more slowly. If the water is moving more slowly, it can be constrained more easily by the electrostatic interactions with the solutes. At lower temperature, you have stronger, more stable solute protein interactions, particularly with hydrophobic groups. And so you have higher hydrophobic hydration enthalpy. When you get to 0 degrees, actually you get a lot of heat released when water starts to be electrostatically attracted via van der Waals interactions to hydrophobic groups. But because it's more strongly attracted, which is possible because it's moving more slowly, you have a higher hydrophobic entropy loss. At higher temperature, the water moves more rapidly. As it's moving more rapidly, it's pulling away more readily and more frequently from the attractive electrostatic interactions with the hydrophobic solute. So on average, those interactions are there to a much lesser extent. You have therefore lower hydrophobic hydration enthalpy, but also a lower hydrophobic effect entropy loss. So if we go from low temperature, we have strong electrostatic interaction, significant negative hydrophobic hydration enthalpy, but it involves then a lot of loss of water entropy. As we increase the temperature, the water starts moving more, therefore there's less of an entropy loss, but you also have less of a hydrophobic hydration enthalpy gain. Okay, so faster solvent motion weakens the interactions and lowers the entropy loss, therefore increasing temperature progressively attenuates the hydrophobic effect. Key point there. As temperature increases, the hydrophobic effect is progressively attenuated. That means hydrophobic hydration enthalpy is reduced, but hydrophobic hydration entropy loss is also reduced, so that both the hydrophobic effect is attenuated as temperature increases. this is one example, important example of a ubiquitous thermodynamic phenomenon called entropy-enthalpy compensation. And this fundamental effect complicates thermodynamics, especially in water solution. So this slide then sort of gets to the heart of the matter, and will be ultimately be where we explain in a qualitative way why it is that lowering temperature can denature proteins and cause protein unfolding, in addition to increasing temperature. So if we, of course, if we think about it for a moment, under this circumstance, this kind of situation, before we go on to 0 degrees, let's just think again about what's going on at 37 degrees. At 37 degrees, we have delta H is greater than T delta S, and so therefore delta H minus T delta S is positive. If we increase T, right, if we increase T, the T delta S term will become higher, eventually will grow. Eventually, as we increase temperature, the magnitude of T delta S will exceed that of the delta H term. And so if we increase temperature in a narrow region, going up, let's say, from 37 degrees, the magnitude of this T delta S term increases until it gets higher. At that point, if T delta S is greater than delta H, delta G of unfolding becomes negative. Basically, as this goes up in the T delta S exceeds in magnitude the delta H, unfolding will become favorable and the protein will spontaneously unfold. And so under this circumstances, things are shown here. Increasing temperature drives a protein unfolding, and that is the thermal denaturation process. So with that in mind, now let's think about what happens when we go to low temperature. Well, as we go to low temperature, the configuration of the folded state of the protein does not really change much. And the configuration of the unfolded state of the protein also does not change much. In the folded state, it's very narrowly constrained conformationally. It's wiggling around, there are bond length vibrations and little bits of bond and bond angle stretching, and a little bit of dihedral angle oscillation, but the protein on the whole is very, very tightly constrained. If you look at the protein at 37 degrees or 50 degrees compared to 0 degrees, it is wiggling around a little more, but it's a very small effect. And so to a first approximation, the change in entropy between the folded and the unfolded state of the protein only varies a very small amount as a function of temperature. And so that means the entropy change upon unfolding in terms of the chain configuration entropy isn't going to change much with temperature. And similarly, the enthalpic interactions of the protein, the packing interactions, the atomic interactions of the protein residues with each other is also not going to change much with temperature. So the intrachain packing enthalpy and the chain configuration entropy changes are not going to be very different at different temperatures. What's going to vary wildly with temperature are the solvent interaction terms. And so when we go down in temperature, the strength of the hydrophobic effect increases, and so the magnitude of both of these solvation-related terms is going to increase a lot. So sorry, that animation went in a slightly weird direction, but hopefully the arrows are clear. When the when the temperature goes down, there is much stronger hydration. The hydration is enthalpically favorable. It reflects favorable electrostatic interactions between the water and the atoms of the protein. And so you get much more favorable hydrophobic hydration enthalpy as the temperature of the water declines. The water moves less. It sort of glues on more tightly to the protein atoms. More electrostatic energy is released. More heat is released due to the favorable electrostatic contractions of the now slower moving water, which is therefore spending more time in association with the protein atoms. Because the water is more constrained, the entropy loss due to the hydration is also going to be greatly increased in magnitude. So now what we want to do, right? But all the solvent terms are going to greatly increase in magnitude because hydration becomes much smaller at lower temperature, much stronger, I'm sorry, at lower temperature. And so now what we want to do is figure out how the relative magnitude of all these offsetting terms is affected by this increase in the magnitude of the solvation enthalpy and entropy effects. As schematized here, right, at 37 degrees, the magnitude of the chain entropy gain was significantly higher than the chain entropy, than the magnitude of the hydrophobic effect solvent entropy loss. But if we increase now the magnitude of the solvent entropy loss, because the hydration is much stronger, the net difference between these two things now changes in magnitude, right? At this temperature, the chain configuration entropy gain is greater than the solvent entropy loss, but at the lower temperature, the chain entropy gain is now lower than the solvent entropy loss. And so now rather than the net gain in entropy we get at 37 degrees, we have a net loss in entropy dominated by the solvent entropy loss at the lower temperature. And so the inverse happens in terms of the enthalpy. At the higher temperature, you were not gaining as much enthalpy, you were not getting as much heat released, or as much energy released due to solvation as you were losing energy doing the work of pulling the protein apart. But at lower temperature, you get much more heat released due to the hydration. And now the relative magnitude of these terms has reversed. And rather than having there's a net enthalpy, there's a positive enthalpy of unfolding at 37 degrees, and that reverses at 0 degrees, and there is a negative enthalpy of unfolding. So at 37 degrees, this whole process is dominated by the amount of energy you need to put in to do the work of pulling the protein-protein interactions apart. At 0 degrees, it's dominated by the very strong enthalpy of hydration in the heat that's released as the solvent molecules glue on more tightly and with less motion to the solvent exposed protein atoms. And so when we go from 37 degrees, where we have both the positive enthalpy and positive entropy of unfolding, due to the very strongly increased magnitude of hydration and the increased magnitude of the hydrophobic effect, we go to 0 degrees. Instead of positive enthalpy and entropy of unfolding, we get negative enthalpy and entropy of unfolding. And so there's been a fundamental reversal in the thermodynamic properties. They're actually driving folding or unfolding, right? Unfolding here is driven by the positive entropy gain of the unfolding at 37 degrees is driven by the positive entropy gain of the polypeptide backbone. But at 0 degrees, instead the constriction of the solvent and the loss of entropy of the solvent dominates, and the sign of the net entropy change upon unfolding changes. Down here, at 37 degrees, we have to put in energy we have to put heat in to pull the protein apart. And the amount of heat we have to put in to pull the protein apart is more than the amount of heat we gain from the increased solvation in the unfolded state. At 0 degrees, it's exactly the opposite. You gain much more enthalpy from the hydration at that temperature than you do. The heat release due to hydration at lower temperature exceeds the amount of heat work you need to do, or the energy you need to put in to pull the polypeptide chain apart, and you get a net. You get a negative delta H, meaning heat is released due to the very strong hydrophobic hydration. And so when you put this all together, when delta H and delta S are both positive, as we increase T, we will drive delta G from being positive. Right here, we have delta H is a bit higher than delta S. T delta S, as we increase T, the magnitude of this will increase, and the delta G, the sign of delta G, which is the difference between these two, is delta H minus T delta S. As this goes up, the sign of delta G is reversed, and delta G of unfolding will become favorable. When we go to low temperature now, when delta H minus T delta S, in the values depicted here, delta H is still greater than T delta S, and so that means the delta G is positive. Unfolding is disfavored. But now, as we lower T, we will lower T delta S. This will shrink, and we'll get to the point where delta H minus T delta S is negative. Then unfolding becomes favorable, and the protein will spontaneously unfold. So the graphs over here then show the sort of sew this all together in a way. This graph is showing the free energy, the net free energy of unfolding as a function of temperature. It is a roughly parabolic plot. The maximum stability for this protein is achieved at 290 degrees. That would be about 17C. That's sort of roughly typical for mesophilic protein. Might be a little bit, most of the proteins for E. coli probably have maximum stability a little bit below that, but this is a reasonable depiction of how these terms work out for the average mesophilic, let's say E. coli or human protein. When delta G is equal to 0, of course, there's a 1-to-1, right? If delta G is equal to 0, the equilibrium constant is 1. We'll have a 1-to-1 ratio of folded and unfolded. At that point, the fraction folded will be 50%, and so this will be the midpoint of the unfolding process. As we go up in temperature up here, we go from fully folded protein to fully unfolded protein. That's thermal denaturation. Down here, as we lower temperature, right? As we lower temperature, the magnitude of this declines. The delta H becomes lower than the T delta S, and delta H minus T delta S will now be negative. Unfolding is favored. Unfold, we go to lower temperature, it unfolds. This is called denaturation. So now the cold denaturation point is well below 0 degrees for most proteins, probably typically around minus 50 C. You can still study this by doing things like putting glycerol in the solution, or, because that if you put solutes in water, they suppress the freezing point. And I don't know if any of you have ever worked with a 50% glycerol solution, but it does not freeze at minus 50. So you can actually do experiments under those conditions. But anyway, for most proteins, the cold denaturation temperature is very low. However, for some proteins, including apomyoglobin, and when I was a postdoc, someone working on the bench exactly opposite mine ended up, sort of struggling with her protein purification for a very long period of time until she sort of accidentally let the cold box warm up, and she found that her protein, which was unstable in the cold, was quite stable at room temperature, which from then on she just purified it at room temperature, and she actually rapidly got a cell paper out of that, and that's something called a major histocompatibility complex protein. It's a very important protein in the human immune system. There was an example of a human protein that actually undergoes cold denaturation around 4C. So it can happen at higher temperatures. It's not common, but you work with enough proteins and you will find one that is cold sensitive. Okay, so what's going on here is, as this graph shows, between 37 degrees, this plot shows, between 37 degrees and 0 degrees, the enthalpy of unfolding goes from being positive to being negative. It completely reverses in sign. The entropy of unfolding goes from being positive to being negative. It also completely unfolds in sign. The stability of the protein comes from, of course, delta G. So what that says is, while net stability is similar to 0 or 37, the fundamental forces stabilizing the native state change. Effectively, the stability comes from the difference between delta G is delta H minus T delta S. It's this small gap in here. So as I said earlier, the net stability of proteins is relatively small compared to enormous differences in the enthalpy and entropy of the folded and unfolded states. And remarkably, the net entropy difference and enthalpy difference in unfolded versus folded changes in sign, not just in magnitude, in sign between high temperature and low temperature. And that change in sign is responsible for the fact that proteins can heat to nature. Right up here, when we increase temperature, we drive this T delta S term higher than delta H, and that makes the delta G for unfolding negative. Down here, as we decrease temperature, that lowers the magnitude of this T delta S, and that also makes the delta G, negative. And so up here, increasing temperature drives you to a negative delta G and drives unfolding. Down here, decreasing temperature leads delta G of unfolding to go negative and drives unfolding as temperature is lowered. And so ultimately, this enormous variation in actually sign reversal in the enthalpy and entropy of the folding process is driven by the enormous temperature-dependent variation in the strength of the hydration of the protein by water, and because there's much more protein exposed to water in the unfolded state, that hydration effect is quite dominant and stronger in terms of the solvent interactions with the unfolded state. And so if you think about everything I've said and look back at the graph, you'll see that unfolding is driven by chain entropy at 37 degrees, right? At 37 degrees, the favorable, right? At 37 degrees, there is energy, enthalpy needs to be put in to do the work of pulling the polypeptide apart, but you're gaining entropy of chain configuration. And so the chain configuration entropy is driving the unfolding at 37 degrees. At 0 degrees, the signs of these things have reversed. And so the net entropy effect is unfavorable because when you unfold, although the polypeptide has increased entropy, the water has lost so much more entropy because of the greater strength of the hydration interactions. And so it's a net unfavorable entropy change when you unfold due to the solvent restriction. But you're getting energetically favorable interactions of the water with the solvent. So there's a net heat release. And then that net heat release corresponds to an entropy gain in the thermal bath. And so hydrophobic hydration, the enthalpy of hydrophobic hydration, is ultimately what is going to be driving the unfolding process at 0 degrees. So there's a total change, in the thermodynamic forces that are driving unfolding between the two temperatures, and it's driven by increased chain entropy at 37 degrees, and it's driven by increased hydrophobic hydration enthalpy at 0 degrees. So this slide, I'm not going to go through it in detail. It just summarizes some of the math. I think to wrap your head around this, you just have to sort of sit here and look at the numbers. But this is just looking back, at the same stuff that I just treated in the last slide. This is just looking at some of the mathematical relationships sort of in the background there. Okay. And really, I've already gone through basically everything that's here. Okay, and so this slide is probably the more important one, and if you wrap your head around this, you're good, and you understand this phenomenon, and you will understand a lot more about biological thermodynamics. And so this slide, I'm not going to read through it in detail because it is what I've just told you, but I think laying it out this way, looking at protein folding and unfolding at either 0 degrees or 50 degrees, I think this is a good way to make sure you understand what's going on. Of course, protein unfolding is exactly the opposite of protein folding. So the signs of the delta H and delta S terms have to be exactly, the magnitude of delta H and the value of delta S and delta H of folding is exactly equal to the negative of delta S and delta H of unfolding, because these are inverse processes. So when near 0 degrees, it turns out folding is what we call endothermic, meaning that we need to put in heat to fold the protein. That's because we're putting in heat to basically strip the solvent, do the work of stripping the solvent molecules off of the exposed protein atoms. So if folding is endothermic, that means unfolding is exothermic. It's because a lot of heat is released when those solvent atoms glue on, as it were, to the protein atoms. And so, of course, at 50 degrees, everything reverses in sign. So if folding is endothermic at 50 degrees, that means unfolding has to be endothermic if folding is endothermic at 0 degrees, that means unfolding has to be endothermic near 50 degrees, based on the schematic shown in the last slide. And so what that means is at 50 degrees, as the last slide shows, unfolding involves an increase in entropy and an increase in enthalpy. At delta H greater than 0, you have to basically put energy in to pull the protein atoms apart from one another, and that work is effectively, the need to do that work opposes the unfolding process, but it is driven by the enormous gain in the entropy of the polypeptide. So near 50 degrees, the polypeptide chain dominates, both the entropy effect of the polypeptide chain, right? The positive entropy of unfolding is because the polypeptide chain gains entropy. The positive enthalpy is because you have to pull apart the atoms the protein atoms interacting with one another in the folded state. So here the chain dominates. Down here, the solvation effect dominates. Unfolding is driven by hydrophobic hydration. That involves an enthalpy or heat, negative enthalpy of heat release, but it is also entropically constraining the solvent. And the sign of delta H and delta S have both completely changed between the two temperatures. So I think that covers all the key points, and it's probably a lot to wrap your minds around in an accelerated time frame. But if you look at this little table and understand that and are comfortable with that, I think you've got it all down, and you know what you need to know, and you are also equipped now to understand much more deeply a lot of thermodynamic phenomena than actually most practicing scientists.